Hello, everybody. In this video, we're going to talk about review part 5, meiosis. So let's take a look. So over here, we have a comparison between mitosis and meiosis. So you can see on the left side that mitosis only allows the cell to divide once, and the daughter cells are identical from the parent cell. In comparison, we have meiosis on the right side where the cell divides once and then divides a second time. And we make 4 cells that are not identical with each other, and they're very different from the parent cell. So you can take a look on the left side where we're counting the chromosome number for a human. So 46 at the very beginning for the parent cell and 46 chromosomes for the daughter cells as well for mitosis, because we're just making more of your skin cells or more of your neurons. You're just making identical cells in this case. Versus on the right side, we have meiosis, where we start with 46, and with 23 after the first division, and 23 again after the second division. So we're going to take a little detailed look at the meiosis process and see how we went from 46 to half the number of chromosomes, which is 23 for a human. So on the top, you can also see the 2N versus 1N. 2N means diploid. Diploid doesn't have anything to do with whether there are sister chromatids or not, but it's really about, are there homologous chromosomes? So as a reminder, homologous chromosomes are a pair of chromosomes, so 2 completely separate chromosomes that are not connected at all normally, and one is from mom and you receive another one from dad. And that's why we call it 2N, because you really have 2 times the number of genes and chromosomes that you would have versus the haploid number, right? Because you have double the number, one from mom and one from dad overall. Versus at the end of meiosis, you really only want 1 copy of each gene. So 1 copy of each type of chromosome as well. So you're dividing up the chromosomes from mom versus dad, and you're only saving 1 of those for each pair of the chromosomes. So we're going to take a look at how that works. There's also a little bit difference between the purpose of mitosis and meiosis, as you can see over here. Meiosis is only for sexual reproduction in eukaryotes. Really, what we're trying to do is you're not going to pass down all your genes to the child. You're only passing down half of your genes. So then the other parent can pass down half of their genes, and then the child can receive one of their set of copies from one parent, the other set from the other copies, and together you make a diploid cell again. And once again, the result of meiosis is you will end up with 4 gametes. So 4 cells, but they are gametes. They're cells for reproduction, and they only have half the number of chromosomes compared to the parent cells. So we'll take a quick look of our overall process. We have prophase, metaphase, anaphase, and telophase, but this is mitosis as we see. We're lining up the sister chromatids, so all the chromosomes just line up in one line, and then the sister chromatids will then separate. And we end up with the same number of chromosomes because, as you can see over here, these are 4 different chromosomes. And after the sister chromatids separate and you divide up the cell, you still have 4 chromosomes in each of the cells. They're just unduplicated chromosomes versus duplicated chromosomes that you have at the beginning and the middle of mitosis. Now, we're going to take a look at meiosis in a little bit, but as a review, what is a homologous chromosome? A little bit of a visual experience over here. So we have a pair of homologous chromosomes. These are unduplicated homologous chromosomes, but either way, we see 1 chromosome from dad and 1 chromosome from mom, and they're completely separated from each other. And you can see that they carry the same type of genes. So for example, the one from dad carries a red hair allele of the hair color gene. Versus on the right side, we have the brown hair allele, so the brown hair version of the DNA that, like this little part of the DNA, allows you to have brown hair, but this one is passed down from mom, and that is totally fine. Both of those genes are the hair color genes, but they don't have to be the same variation or the same allele. So we have a pair of homologous chromosomes right here. After DNA replication, so before meiosis officially takes place, we have DNA replication where every single chromosome, every single DNA, you will get a sister chromatid made by DNA replication. And these 2 identical copies of DNA will be fixed together in the middle, called the centromere. So as you can see on the left side, this is still a DNA that is from the paternal side, from dad. But now, instead of just 1 copy of the DNA, you have 2 copies. And there are 2 sister chromatids. They're completely identical. So this is red hair. This will also be the DNA that codes for red hair. If this is blue eyes over here, the DNA that codes for the eye color gene over here will also be for the trait blue eyes. So we have two identical copies fused together, but this is just one chromosome. Even though there are two chromatids, two DNA, this is one chromosome. And on the right side, we also have one chromosome, and now it's a duplicated chromosome from mom. So we have the two sister chromatids attached right in the middle in the centromere. All right, that's homologous chromosomes. Moving on, what is the difference between somatic cell versus gamete? Let's take a look. A somatic cell has 23 pairs of chromosomes, right? This one kind of looks like they're sister chromatids, but really they're two separate chromosomes because they're not actually connected in the middle. So every one of these, for example, over here, for chromosome number 9, you really have two of those, right? We have two chromosome number 9. And again, why would you have two of them? One is from mom and one is from dad. That's how you end up with a diploid cell, right? It says 2N. It's really 2 times 23. They're 23 individual, like 23 numbers of chromosome, but you have 2 of each kind, so you have 23 pairs of chromosomes, really. So this is a total of 46 chromosomes on the left side. This is a somatic cell. Versus on the right side, we have a gamete. A gamete only has one of each type of chromosome, so this is 23 individual chromosomes rather than the 23 pairs of chromosomes. So instead of having one from mom and one from dad, you only get, like, this might be from grandma, maybe this is from grandpa. Either way, you're only passing down one of each type of chromosome instead of both. Because that's how sexual reproduction works. So on the right side, we have 23 chromosomes, and they're all unduplicated because we don't see any sister chromatids. And this is a gamete. All right, now, diploid and haploid really has nothing to do with sister chromatids at all, because in these two images that we can see on the left versus on the right, both of them are actually diploid cells, right? On the left side, we have the same image as we saw before. There are 23 pairs of chromosomes. There are no sister chromatids, so the DNA is not duplicated yet. Versus on the right side, we still have 23 pairs of chromosomes, but these are 23 pairs of chromosomes that have all gone through DNA replication. So there's still 23 pairs, right? This is 1 and then 2. There are 2 of the chromosome number 1, 2 of the chromosome number 2, but for each DNA, we have a copy of it. So instead of just one DNA right here, right, like one DNA right here, we have a DNA and an identical copy on the right side as well, and they're fixed together in the centromere. So both of these are actually both of these are actually diploid cells, and both of them have 46 chromosomes, and they are both somatic cells. They're not for reproduction purposes at all. All right, moving on. Let's count chromosome numbers again. Mom or dad, you go through meiosis to produce the gametes, sperms and eggs. And then we go through fertilization where half, like the chromosomes from one parent, right, the one copy of each, and the chromosome, one copy of each from dad come together, and you end up with a fertilized egg, and you have doubled the number of chromosomes again. And you go through mitosis to have an entire organism. As a reminder, mitosis is for growth and repair of your body. So you're just making more cells overall. You need to get taller, you need more cells, right? You need more of the regular somatic cells, your body cells, not the gametes. But in terms of chromosome numbers, we start with 46 at the beginning before meiosis. After meiosis, you end up with 23 chromosomes. 23 plus 23, we get 46 chromosomes again, and then you're going through mitosis, so you still have 46. So 46 is really, we say 2 sets of chromosomes, but really the 46 is the right number. You need 46 chromosomes as a human normally, to function as a human being. Okay, moving on, let's take a look at meiosis, breaking down into meiosis 1 versus meiosis 2. So meiosis 1 goes like this. We have interphase, DNA replicated, so DNA uncondensed, really. That's why I'm doing the wiggly finger. So before meiosis, you would have already gone through G1S and G2, but by the time you start meiosis, the DNA is already replicated, right? So like this. However, the DNA, the nuclear envelope is still intact versus this is prophase 1 where the nuclear envelope is breaking down. And then over here, the DNA is uncondensed. So under the microscope, you cannot see the individual chromosomes versus by the time you get to meiosis 1, so prophase 1, the DNA really needs to fix together. Like it needs to wind tightly condensed so that it's easier to separate the 2 copies of the DNA, okay? So we go meiosis 1, right? So interphase, DNA uncondensed. Prophase 1, DNA condensed, homologous chromosomes pair up. Crossing over takes place. Nuclear envelope breaks down. So there are a whole lot that's going on over here. And then we have, you can do metaphase 1, right? Metaphase 1 is where the homologous chromosomes are lining up, right? So one pair, pair. Each pair of homologous chromosomes, one from mom, one from dad, they're going to get separated now. So homologous chromosomes line up at the metaphase place. That's metaphase 1. And then we have anaphase 1A away. Homologous chromosomes separate. That's it. And then we have telophase 1, nuclear envelope reform, DNA uncondensed. And then we have cytokinesis. Okay, so let's take a look. Overall, we have interphase, DNA uncondensed, and the nuclear envelope is still intact. That's how you can tell it's an interphase. Prophase 1, we see the nuclear envelope breaking down using the dotted line. So on the exam, I'll show you these exact pictures so you know what you're looking at. And then we have the X shape, right? The X shape really are the DNAs condensing, and then that's how you're able to actually see the DNA so clearly. And then the homologous chromosomes are pairing up and they're doing the crossing over so that the maternal and the paternal chromosomes are really switching apart. You can see how crossing over works over here. Metaphase 1, homologous chromosomes line up at the metaphase plate, right? M for middle. Anaphase 1, homologous chromosomes separate. Telophase, you see how telophase has 2 nuclear envelopes and they're all in dotted lines, dotted circles. And the cytokinesis is when the cell actually divides. Independent assortment is talking about how the chromosomes kind of line, like each pair, this first pair of chromosomes can line up like this, or it can line up like this. The next pair of chromosomes could be like this, or it could be like this. And the third pair could be like this, or like this. So it's really kind of random, right? We can put dog and cat, and then cat and dog, and then dog and cat again, for example. I don't know if that makes sense, but essentially you're just pairing up the chromosomes, right? They're lining up, but each pair are lining up together and they can be like whichever way. So it really gives you a lot of randomness after meiosis. So that's meiosis 1. We're only dealing with homologous chromosomes. The only part that's about sister chromatid is the crossing over, is homologous chromosomes pairing up, and then the non-sister chromatids cross over. So you don't really need to know that vocabulary for our final exam. Just remember the homologous chromosomes pairing up, crossing over, lining up, separating all homologous chromosomes. And then during meiosis 1, we go from diploid cell to a haploid cell. So based on this picture, this is not a whole human cell. We don't have enough room to draw 46 chromosomes or 23 pairs of chromosomes. So we're just using 4 chromosomes as an example. At the beginning, we have 4 chromosomes, and they're all duplicated, right? We see 2 pairs of homologous chromosomes. So 2n equals to 4, n equals to 2, meaning that there are 2 distinct pairs of chromosomes. So, however, if you're counting chromosomes, you can just count the centromere, 1, 2, 3, and 4. 4 chromosomes. And there are homologous chromosomes, so it's the diploid cell. Now, after meiosis 1, let's say I cut this cell into 2 halves. This is one cell, this is another cell. This one cell now only has 1 and 2 centromeres, so 2 chromosomes now. We cut the chromosome number by half. So it went from diploid to haploid, half the number of chromosomes, right? 4 and then 2. If it's a human, 46, and then this is going to be 23. All right, moving on. So let's take a look at meiosis 2. Now we see 2 separate cells, and each of those cells, as a result of meiosis 1, are going to divide up into individual 2 cells again. And then so one piece of pie, you split it in half, cut it again, you're going to end up with 4 slices of pizzas, right? So you take a cell, cut it in half once, and then cut each of the cells once again. So cutting really 2 times, you're going to end up with 4 cells. So let's take a look at this entire process. Let's just do one at a time. This one is prophase 2. So DNA condenses, nuclear envelope breaks down, just like mitosis, okay? We see sister chromatids, so the 2 halves of the X's, right? 1 and then 2, these are sister chromatids. If you're counting centromeres, 1 and 2, we don't see homologous chromosomes anymore. We separated the homologous chromosomes already, okay? So that's prophase 2. And then we have metaphase 2, right? So instead of 2 X's, we're just having 1 X and 1 line. We say metaphase 2, sister chromatids line up at the metaphase plate. And those are the 2 halves of the DNA, the identical copies. There's a little switcheroo from earlier, the sister chromatids going through sorry, the crossing over taking place, right? So that's why this is a little blue and then this has a little red, but they're still sister chromatids. They're still attached together at the centromere. That's metaphase 2. And then we have anaphase 2. Sister chromatids separate, right? A for away. Telophase 2, DNA uncondensed, nuclear envelope reform. That's why we have the dotted circle. So you're looking for one dotted circle over here versus two dotted circles over here. And then cytokinesis again. So you end up with 4 cells and they're all different from each other. So after meiosis 1, we already, at the very beginning, 2 haploid cells. We already got rid of the homologous chromosomes. We already separated the 4 chromosomes in the image to now 2 chromosomes in each cell. Okay, prophase 2, once again, DNA condenses, and then the nuclear envelope breaks down. Metaphase 2, sister chromatids line up at the metaphase plate. Anaphase 2, sister chromatids separate. Telophase 2, DNA uncondensed, nuclear envelope breaks down, and then cytokinesis again, okay? And then in this case, meiosis 2 really allows a haploid cell at the very beginning to end up with another haploid cell at the very end, 4 haploid cells. Why is it haploid the whole time? Over here, there is no homologous chromosome. We're counting 2 chromosomes, but they are 2 duplicated chromosomes. That's when we have sister chromatids on each chromosome, sister chromatids, but 2 chromosomes. At the end of meiosis 2, we still have 2 chromosomes, but now they are unduplicated chromosomes. So we separated the sister chromatids. We cut the cell into 2 halves, 1 chromosome, 2 chromosomes. It's still 2 chromosomes. So that's meiosis 2, okay? So the whole thing goes, we're not at the whole thing goes yet. Let's compare prophase 1 versus prophase 2. How can you tell? If I give you an image, what are you looking for? If I were to give you a description, what are you looking for? How can you answer the questions correctly? So prophase 1 versus prophase 2. Overall looks exactly the same. The only difference is, do we have pairs, right? If you see 2 paired up together, they're the same size of chromosomes. They look kind of identical, but just in 2 different colors. That's what I'm trying to tell you, that one is from mom and one is from dad. So we see pairs of chromosomes, pair, or 20 pairs, 30 pairs, 10 pairs, 5 pairs, doesn't matter. We see pairs, that's prophase 1. If you don't see pairs, they're just individual chromosomes on their own, but you do see DNA replicating, you do see the sister chromatids, that's prophase 2, okay? If you were to give an actual description in words, this will be homologous chromosomes pair up. That's prophase 1. I don't know what to tell you for prophase 2. Prophase 2 is just prophase 2. There's not much for me to say in a word question, so maybe just an image. Or if you were to put it in the right order, you should be able to do that as well. Let's take a look at metaphase 2. Metaphase 1 and 2. Metaphase 1 have the homologous chromosomes line up at the metaphase plate. These are homologous chromosomes, right? 1 on each side of the cell. These are homologous chromosomes, 1 on each side of the cell. We do this, or you can do this. It doesn't matter. Either way, you have the homologous chromosomes line up and they're going to separate from each other. Versus in metaphase 2, we have sister chromatids line up. We no longer have pairs of chromosomes. We just have 1 line of X's and each half of the X's are going to separate next, right? So 2 file lines versus 1 file line. 2 file lines for metaphase 1, 1 file line for metaphase 2. I don't know how you can remember that backward, but verbally, using words, a word question, if I said homologous chromosomes line up, or if it says independent assortment, those are all metaphase 1. If you see sister chromatid line up, that could be metaphase 2, or it could just be mitosis. Mitosis works just like meiosis 2, okay? Then we have anaphase 1. We still see the X shapes. That's how you know it's anaphase 1, but the X shapes are farther apart from each other. Anaphase 2, you don't see the X shapes anymore. It's just the little lines separating from each other. The sister chromatids are now broken apart. There's no more duplicated chromosomes. They're all unduplicated. The DNAs are separated completely. Okay, so that's anaphase 1 versus anaphase 2. Homologous chromosomes separate versus sister chromatids separate. Then we have telophase 1 versus telophase 2. They look pretty similar again, but the difference is over here we still see the X shapes and over here we don't. And how you can tell telophase versus anaphase is that we see the nuclear envelope reforming. You can't tell if it's reforming or breaking down, but if you see 2 of them, that's reforming. If you see 2 of them here, that's also reforming. If you see 1, that's at the very beginning, that's prophase 1 or 2. Cytokinesis is just cytokinesis. I'm not going to ask you about it. Okay. All right, that's the whole thing. Let's take a look one more time. Interphase, DNA uncondensed. You should do this with me, okay? I know it's, I don't know, if you're at home by yourself or even if you're with a friend, it might be a little weird, but you know what's not weird? Getting 100 on the test. So let's do it. Okay. Interphase. We have interphase DNA uncondensed. Prophase 1. We put it on the side. So prophase 1. DNA condenses, nuclear envelope breaks down, homologous chromosomes pair up. Crossing over takes place. So all of those. Metaphase 1. Homologous chromosomes line up at the metaphase plate. Anaphase 1. Homologous chromosomes separate. Telophase 1. DNA uncondensed, nuclear envelope reform. Cytokinesis. Cytokinesis is just cytokinesis. Prophase 2. DNA condenses, nuclear envelope breaks down. Metaphase 2. Sister chromatids line up at the metaphase plate. Anaphase 2. Sister chromatids separate. Telophase 2. DNA uncondensed, nuclear envelope reform. Cytokinesis. You got it. Okay. Let's do a little practice over here. We see 2 cells. We see there is no more homologous chromosomes. The sister chromatids are separating from each other. This is anaphase 2. This one, we see DNA is uncondensed, so this will just be, you could do interphase, you could do cytokinesis. Actually, this will be cytokinesis because interphase already happened. Or you could try it for telophase, so DNA is uncondensed. You can remember a little brief form. I would say this one is, in my book, cytokinesis because the DNA is fully uncondensed. So if I'm giving you the drawing for the DNA, nuclear envelope breaks down, I'll have the dotted circle. Okay, so let's just put this as cytokinesis. This one will have them paired up. Homologous chromosomes pair up. Metaphase 1. Sister chromatids separate again. This is anaphase 2. Homologous chromosomes separate. Anaphase 1. Cytokinesis over here. Sister chromatids line up at the metaphase plate. Metaphase 2. Homologous chromosomes are paired up and we see nuclear envelope breaks down. Prophase 1. We see 2 circles. We see 2 cells. DNA is uncondensed. DNA is condensed. Nuclear envelope is breaking down. This is prophase 1. And then we have homologous chromosomes separating from each other again. Anaphase 2. Okay. Here is the answer key for the last question that we were just looking at. Well, this was written as telophase 2, 1 or 2. I'm not going to show you these pictures. I'll show you the ones from class, and those will be cytokinesis. But telophase 1 and 2 is also, it's fine, okay? All right, these different phases are, we already talked about it. So I'll show you the answer key in a moment. So you're welcome to just pause it, take a look and see if you can figure out the answers or not. And then after that, you can take a look at the answer key. Okay, benefit of crossing over is to increase genetic diversity. Let's take a look at this last question. A dog has 78 chromosomes in its body cells. So this is a full set. The 2N is 78. So 78 homologous chromosomes. So that will be really 39 pairs, right? So the diploid number, 78, the somatic cell is diploid. So the diploid number is 78. Haploid number is half of the diploid, so that will be 39. And then the number of chromosome in its gamete. Gametes are all haploid, so that will also be 39. Okay, here is the answer. And game, go to Veracross, figure it out. That is all for part 5.